Welcome back, everyone, from your holidays. Happy New Year, and we're glad to have you back with us to the first in our second flight of geomicrobiology seminars. And we hope you all had a good and restful holidays. Just to remind everyone, we will be recording, so if you want to turn your cameras and microphones off, that's fine, and also appreciated if you could hold your questions till the end unless you need clarification, in which case feel free to pipe up. And you can always ask your questions verbally through your microphones or just throw them in the chat if you feel so inclined. And am I missing anything? We're supported, again, just to remind folks, by the MinSoc UK and Geomicrobiology Network UK. And myself and my co-host, Karen Cameron from Glasgow Uni, would just like to welcome you back. And point out that we have an exciting program for the semester that you can see at the Geomicrobiology Network website. So you can get a look at what other talks are coming up. But for today, I'm very pleased to be able to introduce our colleague at Glasgow Uni, Dr. Ciara Keating, who is an expert in anaerobic microbiology and as we were just discussing, really everything from the more basic aspects of methanogenesis and carbon cycling by anaerobes to applied work in anaerobic digestion, and more recently her work in method development to be able to study anaerobes in a more realistic environmental scenario. So and that's what she'll be talking about, the last of those, I suspect, today, looking at her title slide. So, and we're very pleased to welcome her to the Geomicrobiology Network seminar series. And with that, I'll turn over to you, Ciara. Thanks very much.
Okay, hi everyone. So thanks very much for having me here to speak. It's nice to have the opportunity to speak to people from different backgrounds, and I hope you find my talk interesting. It's still a bit of a work in progress, so it's not as if the work is ready for publication just yet, but I think it would be nice to kind of highlight some of the work that we're doing. So the title of my talk is Method Development of an Anaerobic Live Imaging Platform for Industrially Relevant Anaerobic Cells. And really, I'm going to tell you about my work as a postdoc on an EPSRC funded Frontier in Engineering grant, where in my work package, we wanted to apply high throughput anaerobic culturing methodologies to determine the stress response and population variability of key methane microorganisms involved in anaerobic processes. So just to give a little bit of a background about myself, I'm an environmental microbiologist with a BSc and PhD in microbiology, but I've always been driven towards trying to understand complex microbial communities and anaerobes in particular. I don't know why I've chosen the hard road, as we call it. So my PhD was focused on low temperature anaerobic digestion for sustainable sewage treatment. And then in 2016, I moved to Glasgow to work on the Frontier in Engineering grant and also a Global Challenges grant in collaboration with researchers in Thailand. And as you mentioned at the start, I've just now this week moved to a project to understand blackleg disease in potatoes. And it's my ambition to continue in research in applied microbiology, but really focused on innovative tools to manipulate complex microbial communities so that we can harness them for whatever purpose that we need. In the next section, I'll highlight the rationale behind this work, putting it into context, into a global context. So humans produce waste, it's what we do. And as the population continues to increase, particularly in cities, this waste became a public health concern. And one intervention that people started to recognize was that adequate sanitation and clean water were important. And this was largely to do with outbreaks of infectious agents such as cholera in the 1900s, where we had to think past drainage systems and think of waste treatment and the removal of pollutants. And this is where biological waste treatment facilities came to the forefront. And these are methods which harness the activity of a microbial biomass. And as time has gone on, our waste treatment facilities have become more and more complex. And generally, the goal is to treat waste to within discharge requirements at any cost. And our activity has a huge impact on the planet, and this includes our waste treatment practices. And so my research interests have primarily been in anaerobic digestion, which is a sustainable biological waste treatment process where the microorganisms produce methane gas, which we can use as energy. So turning our waste to energy is a win-win. And these engineered systems are underpinned by a complex mixed microbial community. They're typically, so I'm not sure of everybody's background, so this might be a little bit simple, but just to recap is that anaerobic digestion typically has 4 defined stages. So it's not a simple fermentation process, and it requires the sequential and cooperative action of the microbial consortia involved, from the initial hydrolysis to acidogenesis, acetogenesis, and then the final stage where methane is produced by methanogenic archaea. However, it is widely recognized that the true potential of anaerobic digestion is not being met. In full-scale systems, the expected methane yields can vary by as much as 40%, and digestion efficiency is reported to vary from as much as 20 to 70%. And particularly for low-rate systems, it's often highlighted that a waste stream that is consistent in composition and characteristics is best for direct anaerobic treatment. And when you think of a raw waste, it's not often going to be consistent in its composition or characteristics, though it will be from certain defined industrial processes. So often high rate anaerobic digestion does well in these conditions. But many waste streams, such as those with high salt or high sulfide concentrations, are deemed unsuitable or unfavorable for treatment. And yet often when we perform trials in the lab, the outcomes are much more favorable. And there are many reasons for suboptimal performance in the field. Authors have highlighted that operational and maintenance factors play a role. So how often reactors get cleaned or desludged can have an impact, as this can reduce the active space in the reactor. The loading rates and how often a reactor is fed can impact treatment outcomes, and the waste stream itself, and in particular, variability in the influent stream can play a role, and indeed variability ascribed to the microbial community. By its very nature being a biological process, particularly one with various microbial trophic groups, the biology can be unpredictable. And this is kind of a general term for what people say when they, when we don't really have the finer details of the process. And so the microorganisms, they're sensitive to operating conditions and unfavorable compounds in the waste. And we know that the methane producing organisms are the most vulnerable or sensitive to inhibition. And ultimately, these can cause reactor acidification or poor methane yields. Yet we don't have a mechanistic insight into this response at the species level, and we still can't control the microbial community in any kind of targeted way. And in reality, reactor failure is pretty rare. Operators typically are overcautious so as to avoid failure. So this means that the process is not as efficient and we're not getting as much bang for our buck. And in the case of reactor design and operation, we also rely on a great deal of empiricism. So next, I'll give an example of where some assumptions and extrapolations come into play. So biomass growth yield depends on the fraction of energy that's available for cell synthesis. So if we want to gear a reactor towards optimum methane production, we need to consider the acetotrophic, so acetotrophic and hydrogenotrophic methanogens. So these are whether they produce methane from acetate or hydrogen and carbon dioxide. So these are, we often say that these are kind of functioning at the limit, thermodynamic limits. So they don't get a lot of energy from their activity. And so it leads to a small amount of growth yield. So because these are slow growing, we need to retain these in the system. And so if we want to gear towards methane production, then we need to retain these species in a reactor. So this table describes the kinetic parameters for the acetogenic and hydrogenotrophic methanogens. And it shows some really important factors to consider when looking at AD technologies. So looking at the two methanogenic groups, we can see that the observed synthesis fraction varies between the two, and the true yield and rate coefficient vary greatly between the two groups. And so once we get past the initial hydrolysis of a substrate, it's fair to say that methanogenesis is the rate limiting step in the anaerobic digestion reaction. And so when we come to calculate our retention time, so trying to retain the microbial species, for the acetotrophic, we will come out with a value of 4 days, and for the hydrogenotrophic, maybe 0.76 days. And then we add this safety factor. So we multiply by 5, kind of to account for this variability. And so that gives us a retention time of, say, 15 to 30 days. So you can see that all these kind of calculations and assumptions really play into how we operate our reactor and ultimately affect the efficiency of the process. And so an AD system will have billions of microbial cells, and we know that there's a wide range of microbial genera and species that can carry out the same function. So this is termed functional redundancy, and we can easily characterize the composition and function by next generation sequencing and omic technologies. And as microbiologists, we've been doing this for a very long time. However, our investigations into the complex microbial community, typically just snapshots, have not yet been translated to the field. So the ability to control or bioaugment the microbial biomass remains elusive. And perhaps key to answering this is to move away from assumptions and actually characterize the response of these strains to specific stressors in really fine detail. And a source of variation or the response of microbial cells is phenotypic heterogeneity. So this is a phenomenon in which genetically identical individuals will have different characteristics. And it was first characterized in the 1950s, I think, by Novak and Weiner. And so this behavior can happen in any cells, and it's really kind of a characterization of the randomness. So here I'll be talking about prokaryotic cells. And this behavior can occur even if the bacterial cell is grown as a monospecies in well-mixed environments. And this randomness gives rise to collective emergent behaviors that can be observed and described at the macroscopic scale. And so stochastic differentiation or bet hedging, so as part of the population will kind of hedge their bets, and it means that random subpopulations with different phenotypes can appear, and they may provide a selective advantage to survive in fluctuating or unpredictable environments. And so this kind of non-genetic heterogeneity is where cells can switch from different phenotypic states without altering their genomes, either randomly or in response to changes in the environment. And it's more pronounced in kind of dynamic habitats. So a bioreactor or biotechnology would be a good example of this type of environment, and would likely harbor a high rate of phenotypic heterogeneity. And so it's governed by internal or external factors. So for example, internal and intrinsic factors such as the cell cycle state, the age distribution of cells, or stochastic gene expression and metabolic reactions. And so these can be random or they can be in response to environmental pressure. And so most microorganisms live in environments where nutrients are limited or vary, and cells generally respond to these fluctuations by monitoring or adapting their physiological state. And it can help them to kind of survive short-term stresses. External factors such as nutrient availability, chemical gradients, and say pH stress. And the metabolic diversity shown will highly influence growth rates. And so this variation allows microbial species to hedge their bets. So in this case, the population is heterogeneous prior to any change in external condition. And then this subpopulation may have decreased fitness in optimum conditions, but come into their own when a particular stress is applied. And it allows the ability or flexibility to change in response to a particular external cue without alterations in the genome. And so what does this mean for biotechnological processes? So the performance of any bioreactor is kind of cumulative of the interactions at the single cell level. And so variations at this level will profoundly affect the dynamics and productivity of the process. So in monoculture fermentations, this may not be ideal as yields may be lower if cells are not in the same kind of productive state. But in, say, the context of anaerobic digestion, of course, a higher proportion of viable cells will give the best productivity, but a heterogeneous subpopulation within particular active strains may be advantageous in fluctuating conditions. And this is something that we still don't really know in the context of many biotechnological processes, but definitely in the context of anaerobic digestion. And so globally, there's been a shift in how we interrogate microorganisms in the lab. There's also been more of a focus on automation, machine learning, and monitoring microbial communities in kind of simulated conditions that mimic the real environment as much as they can, such as, for example, this build your own soil microfluidic device. And these nanofabricated, so these come from novel microfluidic devices, which allow precise control of fluids. And so you can really precisely control the microenvironment and study in real time the response of the microbial cells. So these offer new opportunities to evaluate phenotypic heterogeneity under realistic bioprocess conditions, where you can scale down and mimic the reactor conditions. And combined with fluorescence, you can kind of track specific genes and really carry out high throughput, deeper scale experiments. And so I believe that using high throughput, highly controlled experiments with key species in the AD process are key for us to eventually control or understand the biology at a finer scale. But when we think of the tools that we have in our anaerobic culturing toolkit, they're not great. A lot of the time we use bulk or averaged community data, and we often have to use destructive sampling. And we can't observe cell-to-cell interactions or examine cells in situ environments. And for anaerobic organisms, it's challenging because we have to create that anoxic environment for the microbial cells. And though authors have yielded incredible insights using these methods, the performance of individuals within the populations is masked. And I think that when we come to looking at our anaerobic digestion technology, we really need to understand what each individual species is doing to be able to kind of produce better models or infer a better process outcome. And so with this in mind, the aims of this work were to develop a method for anaerobic live imaging growth of anaerobic cells, test the ability to cultivate industrially relevant strict anaerobes, and uncover growth rate heterogeneity within single populations. So as this was bringing together a lot of new techniques, the methodology was a real technical challenge. A little bit like going down the rabbit hole, as each part required optimization. I haven't included everything here, as I don't want to kind of bore you on the first week back. But I'll try to cover as much as possible. But if you're interested to learn more, you can ask after or send me an email. So what we required for the platform development was strict anaerobic conditions, the ability to control temperature, accurate fluid delivery, a device for growing and holding the cells, and then the capacity to visualize, so to distinguish and track individual cells growing in real time and potentially over long timescales, because anybody that's worked with anaerobic organisms know that their growth rates are slower typically. So we had I grew a panel of anaerobic microorganisms. I've added one or two more methanogens to this, but really we wanted to kind of get a feel for how many of these organisms might grow and in what kind of devices. So, and we also looked at syntrophic bacteria, such as Syntrophobacter willeni or Desulfovibrio vulgaris, though we didn't carry out any co-culture experiments in the end. So for the initial device fabrication, so the microfluidic device was manufactured using polydimethyl siloxane, so PDMS. And so this is a really kind of handy material for fabrication of these devices. It's relatively cheap and can be fabricated relatively easy. We were lucky to have collaborators in the School of Engineering who were already highly skilled in microfluidic device fabrication. So initially we started using a device design that they had already prepared. And so within this, you have two channels where the flow of medium, you have diffusion of the medium into this culture well, which is held up by support pillars. And this is really a chemostatic device. So the cells are held within this culture chamber, Waste is allowed to exit through here, but this height here should be small enough so that the cells can't escape, so that you can track them growing over time. So that's just an example of the kind of the full scale of the device. So you have your two influent channels and the two effluent channels, and then the 6 culture chambers. And this just shows the kind of filling of the device with the growth medium. So that was the microfluidic devices. So next we had to think about how to monitor the cells and how to maintain anaerobic conditions. So our collaborators had used the system with E. coli. So in this case, the device was mounted onto a microscope just out in the open. So obviously for our anaerobic cells, that wasn't going to be enough. So we looked initially to a methodology from Steinhaus et al. From 2007, where they took a methanogenic species and had a microfluidic bioreactor, which was in a sealed gasket chamber, and sparged it with gas whenever they were monitoring on the microscope. And so this would maintain anaerobic conditions. So one of the first things we encountered was that the microscope sees everything. So anoxic media is complex. So this image here is just the medium without any cells. So optimization was required to produce a clear medium. So if we think of kind of any kind of monitoring with the microscope, with all of these particles, we're not going to get good images, and it's going to be harder for the image analysis to actually distinguish the cells. So we found by by augmenting the reducing agents and changing some of the media components, we were able to get a clear medium. But then optimization was required to use a medium that was providing the quickest growth rates, because the longer you have your device running on the microscope, it gets harder and harder to kind of to work with, basically. And our collaborators had identified that. So they had worked with E. coli, so with doubling times of, say, 20 minutes, whereas we're looking at doubling times of, so this was Desulfovibrio vulgaris, so this is in hours here. So we wanted to get the quickest growth rates, basically. So another thing that I had considered was that we're looking at phenotypic heterogeneity, so we want to see the differences in the individuals. So that means that in our anaerobic media preparation, our media should be as consistent as possible. And when making anaerobic medium, the method that we were using previously is where you would have everything in, you would prepare the medium and then prepare into the anaerobic vials, autoclave the vials, and then add the different components to each of the vials afterwards. So that creates a lot of variability between your different vials. So you can't say that these are the same. So we wanted to kind of eliminate any kind of as much variation between the different sets of media as we could. So using this is a whittle setup. So basically everything gets put into this flask, the different media components, and then this is autoclaved and sparged. Everything's added, the reducing agents to this flask, and then aliquoted into the vials. So each vial should be the same. But also we thought about the setup where we sparge the microfluidic device just out in the open on the microscope, and combined with our reducing agents, we needed to get more anaerobic conditions, basically. So we needed strict anaerobic conditions of 0 to 5 ppm. So I redesigned the experiment and actually decided to put everything inside of an anaerobic incubator so that we could maintain strict anaerobic conditions. So just to give some of the results. So this is the platform. So tentatively called it the Anaerogrow platform. So this is taking an anaerobic incubator system, which is capable of going to 37 degrees, embedded with a automated microscope, microfluidic pumps, and microfluidic devices can be then mounted onto the microscope, which is controlled using a computer outside of the chamber. So working within the chamber itself is quite challenging, particularly working with really fragile microfluidic devices, which are the microfluidic device is mounted onto a really thin cover slip, which is really fragile. This is just an example of working within the chamber. So it's quite a tight enclosed space. We've played around with different types of devices. So we had the devices that our collaborators made, and also tested some devices that were available to purchase to kind of give the full range of what was possible within the system. So then we wanted to test the ability to cultivate industrially relevant strict anaerobes. So this is Desulfovibrio vulgaris in the device design that our collaborators made. So this is just an example of the cells in real time, just taking a video. But really what we can do is track the growth of the single cells over time, which I'll explain in a bit more detail. But this was a really good result. We could see that the cells were highly active and that we were getting growth within kind of a really short lag phase. I also tested some methanogens. So this was Methanococcus maripaludis, which is a hydrogenotrophic methanogen. So I saturated medium with hydrogen because we were unsure how to feed gaseous substrate to the device inside the chamber. And obviously hydrogen is flammable. So I guess the image of this isn't ideal, but we did get growth in 5 days, completely covering the device with the Methanococcus cells. The challenge with this is that the chamber height was smaller, or was too high, so cells initially washed out into the flow channel and then grew down. So really the device dimensions are really important. Recently, I did some analysis with Methanobacterium formicicum, and these were quite active. I'm still analyzing the results from this to actually calculate the growth rates. One of the challenges with the methanogens is that they like to form biofilms. So this was in a commercially available culture well, so the cells were not constricted height-wise. So we got so there was a lot of biofilm production, and the cells were free to move around. We've also tried Methanosarcina barkeri and methanococcides methyluteins, and I'm currently analyzing the images from the last run with these. So hopefully we'll have kind of a more detailed analysis. So with the methanococcides methyluteins, I tested growth in methanol, but also in trimethylamine. And I'm going to do a comparison of the growth rates in the different culture medium. And so we then wanted to uncover the growth rate heterogeneity in a clonal population. So looking at the individual cell growth rates under pH variation. So from pH to pH 6, and to test the distribution of the growth rates per individual cell, and see how the distributions change with an applied pH stress. So for the experiment with Desulfovibrio vulgaris, I tested, this was from an older experiment, I tested pH 7 and a pH shift just to pick pH 6.5. So in order to calculate the individual cell growth rates, the time-lapse images are taken. So starting out with our time 0, identifying the cells that we're going to track, and then the area of that cell is calculated, and then as that cell grows and divides, the increase in its cell area is calculated. And then we calculate how this increases over time to give us the slope and the specific growth rate. And so in this case, so each bar is the growth rate of one cell. So I've only shown 20 cells here. And so it's only preliminary findings. But here we can see that, so this is our doubling time. So at pH 7, we have more variation in our growth rates. And whereas at pH 6.5, we seem to have more uniform doubling time of the cells, although more data or more cells are needed to really say for sure. So I'll come on to describe that in a minute. But what I did notice was that only 20% of the population were actively growing. So this is an interesting finding, in that we make all these assumptions about, say, the community average, but only 20% of the population may be actively growing at any time. So one of the limitations of this device was that the Desulfovibrio cells started to form a biofilm, which kind of limited our ability to track individual cells. So we've had a PhD student, Io Ogundero, who has really kind of taken the lead on designing new microfluidic devices so that we can see here, so that we can constrict the chamber height to give us a monolayer of growth. So that has been quite challenging, I guess. So it's not so simple as to just reduce the height, because here we're at 0.6, I think it was, and the lower you go, the harder the fabrication process is. So IO has just recently created masters for something more like a mother machine type device, where you capture one cell and then track the growth of that one cell. So hopefully that will be a little bit easier to work with. So with COVID-19 disruptions in the past year, we've been a little bit tricky to get that off the ground, but hopefully have a device soon. And I guess with that, so to conclude, we've developed an anaerobic incubation and microscopy monitoring platform for non-destructive growth analysis of anaerobic cells. It's still a work in progress, but I guess my real take-home message is that we need to culture more, and we need to culture better in terms of anaerobic culturing, and really kind of embrace the possibilities of what we can do. There's a huge range of options, from microwell devices that you can purchase. There's and different systems. So really, there are options available to be able to do high throughput analysis for key anaerobes. And hopefully this will help us to harness the true potential of these species, but also in terms of kind of fundamental microbiology. There's a lot of questions that still need answered in terms of the methanogenic archaea and different important anaerobes. So we did observe heterogeneous growth rates in the individual cells in Desulfovibrio vulgaris. And this heterogeneity appeared to reduce with stress, although we need to test this in kind of more devices to know this for sure. Before Christmas, I ran experiments with Desulfovibrio in little micro plates, which seemed to have given good resolution of single cells across multiple wells. So the image analysis is quite intensive, so it'll be a while until we have the results of that. And I guess the other thing is that investment is needed. I think it goes beyond the work of one lab or one group. And really, we need to kind of have investment into kind of anaerobic fluorescent markers, and really kind of try to move forward the toolkit that we have. So with that, I'd like to thank you for your attention, and to thank everybody at the Water and Environment Group and all my collaborators. Okay.
Thanks very much, Kiara. It's hard yards with, I think, some of the work you've done clearly, and I think there's the applications and the implications are amazing. I mean, you could imagine the same sorts of non-averaging issues being relevant to many natural environments that and many other industrial environments as well. It makes me terrified to think about how we draw conclusions from. I mean, I never want to use a batch, a Hungate tube or bottle again, I think. But, and by the way, I should apologize for, sorry, with my holiday brain on, I neglected to mention your academic track record. So I thank you for including that on your second slide, which worked out fortuitously.
Thank you. Yeah, it was a shameless pitch because I am hoping to get a fellowship or a
perfectly
lectureship
timed.
at some point.
Perfectly timed. Great.
Yeah,
so
it was a good opportunity.
With that, I'll open up for questions. I know I've got some, but I'll defer to hear any questions from the audience.
I've got a couple of quick questions. I guess the first is just out of interest from your introduction. I'd never thought about that not all treatment plants ought to be used in the same way. Does that mean that if you're working with different human communities with different diets, you need to consider that with the way that you treat them? Is that what you were saying about the sodium component?
What was that part?
Well, at the start, I thought you said like if the like sodium levels can affect the way that you deal with waste treatment. Does that mean
yes, so,
different diets that you have to do?
So I meant with high sulfide concentrations.
Oh, okay.
Like sodium sulfide, which can impact the methanogens. So typically your methanogens will be inhibited by high sulfide concentrations. So one challenge is, say, like tannery wastewater, which will be kind of high acid, kind of high sulfide concentrations. The application of anaerobic digestion to that kind of wastewater isn't really done. It's typically kind of not suitable. But I think if we actually kind of move towards engineering the methanogens to be more robust, because there are methanogens that can survive higher concentrations.
The second question was just a quick one on the software that you use to track and measure those cells. Have you worked with a few, and have you found one that you really like? Because I've struggled in that area.
Oh my God, it's a struggle. So we did have a postdoc who was employed to kind of do the image analysis, but with how things worked out with delays in the device designs, she's now finished, and I had initially done the image analysis for the first kind of round of device designs, and I did it manually using kind of ImageJ pretty much. So there's Cell Profiler and some other softwares, but initially I've been doing things by hand. And that's why I didn't show that many cells, because it took so long to analyze just a few cells, and then that device wasn't kind of optimized. But we are hoping to use kind of a MATLAB pipeline, but to be able to kind of automate that part of it. But you need to be really sure of what you're putting in, and because we're not using fluorescence, it's harder to distinguish kind of what is a cell or what is a pillar.
Okay. Yeah. Thanks.
Can I ask, why is it that you get some biofilm forming along one side, but it looks like you've got free swimming cells on the other, unless I misunderstood that?
Yeah.
Why not biofilm everywhere?
Yeah, so with the way I think it's to do with the dimensions of the device. Don't know if I can get this up.
And did I understand you correctly that you said that you're going to change the height of you're going to basically lower the ceiling and make the cells go into a little tunnel? Is that right?
Yeah, so that had, I can't seem to.
Do you think they'll like it in there?
That had its own challenges. We did, poor Io, the PhD student, we've tried a lot of different things. So initially with the smaller design, cells gathered outside of the hole and formed a biofilm outside. They didn't want to go into the tight space. So that's why we've moved towards the mother machine type device,
Right.
because that should just trap one cell, and then you track the growth of that generation, basically. But in terms of the device design, so there's two flow channels, and then you have your culture chamber. And then there is this step down, like, so it goes from 0.6 to say 0.3. And that's where the waste gets removed, but the cells don't get removed. But I think there has to be some kind of force there that the cells seem to like to get up to that point, and then they grow down.
Right. Okay.
Yeah. So
Would you say
it's like they get towards the outlet for the waste, and then kind of grow back down the way.
So I couldn't quite decide whether it seemed as though this microfluidics approach is a good approach to studying biofilms, or do you think biofilms, or do you think the biofilms grow somewhat and unpredictably or inconsistently in that type of geometry? I was trying to sort of get a sense for that.
Yeah. So I think it was hard in the geometry that we were working with. There's other kind of, with mother machine type devices and things, I think you can limit biofilm development. But I think you're always gonna get variation in the cells. So with Desulfovibrio, it's not a simple circle shape. So depending on what way it enters, it's going to be restricted in a different way.
Yes, good point.
So I think it was just another kind of variable that is a little bit harder to consider. But I think if, like, going forward, I think looking at fluorescence and then maybe just excluding the cells that are forming a biofilm. But it's something in reading the kind of literature on microfluidic in devices in kind of aerobic settings doesn't seem to be as big a problem. But maybe that makes sense.
Yeah, having to work in an anaerobic environment adds a whole nother layer of complexity onto it, doesn't it? Do you?
Yeah.
I just want to open it up again for any questions from the audience. I've got like a list, but I don't want to hog up the time. So does anyone want to ask Kiara a question? If not, if you don't have one ready now, if you want to ask a question later, I'm sure she'd be happy to receive your email, or if you think of something and want to throw it in the chat, that's great too. But you showed, if I could just ask one last, you showed something about a build your own soil paper from that ISME J article. So do you think these devices or I guess I should say, how good do you think these devices might be at simulating more realistic sort of, three-phase environments with particles and fluids and of both liquid and gaseous natures? Is this the way that we can do some more realistic work in terms of soil matrices in the lab and still be able to observe things?
Yeah, I think so. It's highly technical. I know, so that there's that ISME paper, but there's also, I think, Toby Keir's group, where they're really able to get some really nice kind of imaging in situ environments. And I think it's easier for labs that kind of are highly specialized in microscopy already, maybe.
Right. Okay.
But I think you can simulate kind of complex conditions, but I guess now I'm maybe thinking more towards kind of building more synthetic communities first, you know, where you can have your different cells in, say, the different like trophic groups and have them share metabolites as a means to kind of monitor kind of the relationship between them.
I love this bet hedging concept. I hadn't thought of it in terms of that before, but I'm going to remember that. That's really in my brain.
Yeah. I think because that was something that I was kind of like, oh, that's really clever. But
it's an interesting way to think about it.
And when I was reading about that, it was mostly geared towards kind of the negative impacts sometimes, because of, say, that really impacts in kind of monoculture fermentations. So they can get low productivity because all the cells are kind of maybe waiting around to see what happens. But in anaerobic digestion, that might actually be an advantage.
Yeah, right. Wow.
And we still don't really know like how the cells respond to kind of all these kind of fluctuations.
Well, thanks again for kicking us off today in our second seminar series. We really appreciate you taking the time out from playing in the snow to be here.
Yeah.
And thanks to everyone for coming out as well. And just a heads up for next week, we have Professor John Lloyd from University of Manchester on board with us. And you can go to the Geomicrobiology Network website to look at the schedule for the rest of the term. And I think that's all from us. So thanks again for coming out, everyone. We'll see you next week, hopefully. And thanks, Kiara, for your talk today. It was really, really interesting and